Hi, welcome to our seminar on saponins, a possible answer to metabolic syndrome. My name is Nathaniel Price, and I will be your facilitator as we discuss the potential of plant-derived saponins in the intervention of metabolic syndrome. By the conclusion of this presentation, you are expected to gain a clearer comprehension of what saponins entail. You will also be able to delineate the physical and chemical properties of saponins. You will also be given a concise overview of the biological activities associated with saponins, and you will scrutinize the potential of saponins as an intervention in metabolic syndrome. We are going to be focusing particularly on obesity and dyslipidemia. Hopefully, you will acquire new insights throughout this presentation.
Have you ever wondered what saponins are? Well, today we uncover the mystery of these compounds and explore their fascinating world. So, what exactly are saponins? Picture this: saponins are a class of chemical compounds naturally occurring in a wide variety of plants. They are widely distributed in all cells of legume plants, which are staple items of the human diet. Saponins derive their name from their ability to form a soap-like foam when shaken in water. Quite intriguing, isn't it? But what makes saponins truly captivating is their complex structure. They are glycosides, meaning they have a sugar molecule attached to a non-sugar molecule, known as an aglycone or saponin. This combination creates a wide range of saponins, each with unique properties. Now, you might be wondering, why should I care about these saponins? Here's why. These compounds are no ordinary ones. They pack a punch with an array of potential health benefits. For instance, some saponins have shown promise in supporting heart health. They can potentially bind to cholesterol in the gut, preventing its absorption and thus promoting heart health. Moreover, other saponins may possess anti-inflammatory properties. They could help to soothe inflammation in the body, which is crucial for maintaining general wellness. Saponins also have the potential to boost the immune system, thanks to their ability to stimulate antibody production. To summarize, saponins are natural compounds found in many plants with a unique soap-like property due to their complex glycoside structure. Their potential health benefits range from supporting heart health and possessing anti-inflammatory properties to boosting the immune system. In our journey of understanding saponins, let's take a closer look at their composition. Saponins are unique in their structure, generally consisting of either a sterol or a triterpene unit. Now you might wonder, what on earth are sterols and triterpenes? Well, these are types of molecules that play key roles in the biological world. Sterols, for instance, are vital components of cell membranes, while triterpenes are involved in a variety of biological activities. This unique composition of saponins gives them their fascinating properties and potential health benefits. It's like each saponin molecule is a tiny factory, producing benefits for our bodies. When we consume foods containing saponins, we're not just eating. We're engaging in a complex chemical interaction with nature, one that has the potential to support our health in various ways. So the next time you bite into a crisp apple or enjoy a bowl of oats, remember, you're not just eating food. You're partaking in a beautifully complex chemical dance, one that's been choreographed by nature to potentially benefit your health. Isn't that a wonderful thought? Stay tuned as we continue to delve deeper into the fascinating world of saponins.
Now, what are saponins? Saponins are classified as glycosides, and they are compounds that are widely distributed primarily in plants, but you may find some in bacteria or lower marine animals. This heterogeneous group of compounds is extensively found in both wild and cultivated plants, especially in legumes that are fundamental components of the human diet. So you may find legumes like soybean, chickpeas, kidney beans as having sources of saponins. Plant-derived saponins have been incorporated into various folk medicines due to their diverse health-promoting properties. Given their potential to inhibit pancreatic lipase, there's a growing interest in utilizing them for the treatment of obesity and related disorders, a topic we will explore further in this presentation. Fun fact, the saponin content varies among legumes. For instance, each kilo of dry matter of chickpeas may contain 2.3 grams of saponin, while kidney beans could have up to 3.5 grams per kilogram. So let's look at the chemical and physical properties of saponins. So the structure of saponins comprises a sugar molecule, or a sugar moiety, I should say, and it's typically galactose or glucose or glucuronic acid or xylose, etc. Right? And that is normally attached to a hydrophobic, a glycone or a saponin structure, as shown in the figure there to your left. So you see the sugar chain that's attached to the aglycone structure of the saponin molecule. So when we look at the structure of saponins, they can be divided into two groups, or they're classified in two groupings. You have the triterpenoid or the steroid, and these are found prevalent in terrestrial higher plants as well as some marine organisms like starfish or sea cucumbers, as we had discussed earlier. The triterpenoid saponins are predominantly found in cultivated crops such as legumes, and we spoke about that. There can be either soybeans, beans, peas, chickpeas, etc., as well as in alliums, tea, spinach, sugar beet, quinoa, licorice, sunflower, horse chestnut, and ginseng. On the other hand, the steroid saponins are common in plants used for their herbal or health-promoting properties and can be found in oats, capsicum, peppers, tomato seeds, alliums, asparagus, yam, fenugreek, yucca, and ginseng. The extensively studied group of triterpenoid saponins is obtained from the Quiaha saponaria tree that is native to the Andes region. Indigenous peoples are known to peel off the bark extract of the saponin. They extract it in water and then they use it as a shampooing agent. Shamans also utilize it for curing diseases, as stated by Francis et al. In 2002. The yucca plant, which is a herbaceous plant of the lily family, native to the deserts of the southwestern United States and northern Mexico, serves as the most common commercial source of steroid saponins and is used a lot in folklore medicine as an anti-arthritic and anti-inflammatory agent. Here we have some pictures depicting marine animals, sea cucumber and starfish, that contain saponins, as well as some plants. So we all know soybean, kidney bean, horse chestnut, and ginseng. These are plants that also contain saponins. Saponins are believed to have uses in plants, and these uses are not very well understood. However, the literature has documented that saponins are known to be the plant's defense mechanisms, offering antimicrobial properties, inhibition of mold growth, and protection from insect attack. And we all know that the saponins are secondary metabolites in plants, which usually serve as the protection for the plant. Saponins are considered natural surfactants. Their excellent emulsification, solubilization, and foaming properties make them suitable for many industrial applications such as pharmaceuticals, cosmetics, and beverage industries. According to Francis et al., saponins have also been found to have biological effects in animals, such as effects on the immune system, reproduction, cholesterol metabolism, hypoglycemic activity, cytotoxic effects on malignant cells, to name a few. These effects have sparked interest by scientists for the potential pharmacological properties Plants rich in saponins have been used in folklore remedies in traditional Chinese medicines, which have been documented in the literature, especially in the area of obesity, which is the main focus of our presentation. Now let us look at some of these biological activities of saponins. Effects on cell membranes. The observed biological activities of saponins have been linked to their action on membranes. Francis et al. 2002 indicated that saponins have been shown to have a lytic action on erythrocyte membranes, which have been used in their detection during laboratory activities. This pronounced hemolytic activity can be attributed to the interaction of the saponins with cholesterol in the erythrocyte membrane, due to the affinity of the iglocone portion of the structure for the membrane sterols, specifically the cholesterol that's found in the membrane that forms insoluble complexes. Numerous studies have underscored the impact of saponins on membranes, presenting potential benefits, particularly in the area of cancer research. In their 2014 review exploring effects on artificial and biological membranes and potential implications for red blood cells and cancer cells, Laurent et al. Highlighted the examination of saponin interactions with membranes in silico, artificial models, erythrocytes, and cancer cells. These interactions were found to potentially reduce resistance to chemotherapeutic agents or hinder viral infections. The authors also noted the potential of saponins to activate death receptors in cancer cells and other RAP-dependent properties. Additionally, the authors suggested a correlation between saponin-induced cell death and their permeabilizing activity. Beyond causing membrane lysis, saponins are thought to possess the capability to induce apoptosis and autophagy, or impede the cell cycle and cell proliferation, which is what we are looking for when we are talking about cancer research. Saponins have been shown to exhibit hypoglycemic effects, and these effects have been observed in saponins derived from plants such as the philodendron cortex, also known as hongbai in Chinese or abaku in Japanese, the Aurelia cortex, the Calendula officinalis, and fenugreek. That has been also reported in the literature that these have also exhibited hypoglycemic effects. A study found that higher plasma insulin levels in Western rats was believed to be the result from the stimulation of the beta cells in the pancreas. Additionally, there is evidence suggesting that the hypoglycemic activity may stem from the suppression of glucose transfer from the stomach to the small intestine and the inhibition of glucose transport across the membrane of the small intestine. So it is inhibiting glucose from being absorbed from the GIT into the cells, which will cause a lowering of the glucose levels in the blood. Now, studies have shown that saponin has the capacity to lower cholesterol, both in animal and human subjects. The consumption of saponin-rich foods containing soya beans, chickpeas, and so on were seen to result in an increased excretion in bile salts. The saponins are believed to interact with the sterols in the intestinal lumen, forming micelles, thereby preventing the cholesterol from being absorbed. Consequently, saponin possess the potential to inhibit the absorption of both dietary and endogenous cholesterol entering the intestinal lumen through bile or from the intestine, which is going to, of course, lead to a reduction in blood cholesterol levels, even in the presence of low cholesterol diets. Examples of saponins derived from plants that exhibit inhibition of intestinal cholesterol absorption and reduction in blood cholesterol levels in animal models include steroid glycosides like digitonin, tomatine, and sarsasaponin, as well as the triterpenoid glycosides found in alfalfa, soy, quiaja, saponaria, gypsophilia, and ginseng. None of this you can find in research done by Harwood et al. 1993. Saponin-based adjuvants can activate the cell-mediated immune system and boost antibody production even at low doses. In vitro studies further indicated that both crude mixtures and purified compounds of Kiaha and other saponins have been shown to augment immune cell proliferation. So saponins can boost the immune system. Other biological activities of saponins include antioxidant effects, cytostatic effects on malignant cells, and antifungal effects. Let's look at antioxidant effects of saponins. Saponins bind to cholesterol and prevent its oxidation in the colon. In living tissue, oxygen radicals attack lipids, removing the doubly allylic hydrogen from polyunsaturated lipids and promoting lipid peroxidation, which leads to cell damage. Now, DDMP saponins are a major saponin group widely distributed in legumes such as peas, barrel medic, chickpeas, and soybeans. And they have found that DDMP saponins play a crucial role in preventing lipid peroxidation, or DNA degeneration, and protecting proteins from free radical attacks. So that's how saponins, especially the DDMP, and I'll put the long name on the screen for you, that's how they exert their antioxidant activity. Now let's look at the cytostatic effects on malignant cells. Saponins, isolated from various plant and animal sources, have demonstrated the ability to inhibit the growth of cancer cells in vitro. The activities of saponins against malignant cells have garnered significant interest, particularly in the field of cancer therapy. As mentioned before, numerous studies have indicated that saponins exhibit anti-cancer activities, which includes anti-proliferation of cells, anti-metastasis, anti-angiogenesis, and the reversal of multi-drug resistance, which is a big problem now in cancer therapy. These effects arise from the induction of apoptosis, promotion of cell differentiation, immune modulatory effects, binding of bile acids, and alleviation of carcinogen-induced cell proliferation. Now, it's very crucial to emphasize that these observed anti-cancer activity of saponins is intricately linked to the structural components, including the I-glycone moiety, the length and linkage of the glycosidic chain, whether there is a presence of functional carboxylic group on the I-glycone chain, the quantity of sugar molecules, hydroxyl groups, the position and stereoselectivity of the hydroxyl group, and the type of sugar molecule on the glycine chain. For example, in this 2016 article titled Saponins from Chinese Medicines as Anti-Cancer Agents, Zhou et al. Reported that genosacide RG3 exhibited therapeutic effects both in vitro and in vivo against a spectrum of tumors, and these tumors may include leukemia, lung cancer, esophageal carcinoma, gastric cancer, colon cancer as well, breast cancer, ovarian cancer, prostate cancer, which is quite promising when you think about it. Lastly, let's look at the antifungal activity. It has been suggested that the primary mechanism driving the antifungal effects of saponins is pretty much linked to their interaction with membrane sterols, and that is linked or correspond the activities corresponding to the structural components of the saponins. So saponins with a branched chain trisaccharide moiety lacking oxygen-containing groups at the C2 and C12 position displayed antifungal activity, while those with 2B hydroxyl or 12 keto groups showed minimal or no activity. Saponins containing a disaccharide moiety exhibited relatively low activity, and the aglycones, or the bidesmodic furostenol saponins, demonstrated no activity at all. Now, with all the activity of saponins discussed, do we think that they have the potential to treat or be a treatment intervention for metabolic syndrome? Let's explore a little bit more. Now, obesity has emerged as a significant public health concern globally and is now considered a widespread epidemic. Characterized as a chronic metabolic disease resulting from an imbalance between energy intake and expenditure, obesity leads to the excessive storage of energy as triglycerides in adipose tissue. Now, obesity is a risk factor for various chronic illnesses, including cardiovascular disease, hypertension, type 2 diabetes, cancer, osteoarthritis, asthma, etc. Researchers have explored different drug molecules for obesity treatment. While there have been different pharmaceutical drugs that have been produced in the market for obesity control, many have been withdrawn because of their reported serious adverse events. The initial drug introduced for weight loss, dimetrophenol, in 1930s was quickly replaced by the amphetamines, which were chosen because of their ability to suppress the appetite. However, even these drugs were eventually banned by the Food and Drug Administration due to their severe side effects. Consequently, there is a significant interest in identifying a natural and safe approach to weight loss. Saponins with reported pharmacologic activities such as anti-inflammatory, immunostimulant, hypocholesteremic, hypoglycemic, cytotoxic effects, they are being now considered as potential compounds for obesity treatment. And this has sparked great interest, or increasing interest, in investigating natural products as safe and effective tools for maintaining a healthy body weight. Now, Morelli et al. 2016 conducted a review exploring the impact of saponins derived from medicinal plants on lipid metabolism and their potential health benefit in treating obesity. Now, you'll see that journal article, the abstract, to the left of your screen. The activity of this seminar, therefore, is for us to examine and discuss the outcomes of this review paper, focusing on intriguing findings related to the suggested mechanism of action and the potential advantages of utilizing saponins from plant sources in the context of obesity treatment. I will briefly give a background on what the authors presented in the paper on recent research findings as it relates to anti-obesity therapeutic potential of these plant-derived saponins, setting the stage for a deeper exploration of their effects and implications. So I would like very much for you to read this article, make your notes, and focus on the points that I highlighted just now. Okay, so let me give you a brief background on what the authors presented in this paper. They presented on lipase inhibition, adipogenesis inhibition, appetite regulation, and lipid metabolism. In terms of lipase inhibition, they discovered that some researchers have found that the balloon flower, let me see if I can pronounce this, Platycodon grandiflorum, they found that it was effective in hindering the absorption of dietary fat by inhibiting pancreatic lipase activity. So that is a possible mechanism of action. In terms of adipogenesis inhibition, just a little background here is that when we consume a meal, say you go and you buy a burger and you eat it, the surplus energy, that means the energy that you're not using at the time, is going to be typically stored in the adipocyte cytoplasm as fully differentiated adipocytes through the process of lipogenesis. And this whole process is known as adipogenesis. This regulated process involves triglycerides. However, severe obesity is marked by an increased number of adipocytes resulting from coordinated transcriptional and cellular events during pre-adipocyte differentiation. In other words, you're going to pile on the fat. So if you are able to control adipogenesis with its various potential regulators, it could serve as an effective tool against piling on the fat and therefore reducing the chances of obesity. In a study, 2 types of saponins were extracted and they exhibited anti-obesity potential by reducing triglyceride, total cholesterol, low-density lipoprotein cholesterol levels in an obese Zucker fatty diabetic rat model. So this is proven in animal models. So they're thinking maybe we can now transfer this in humans. Another area that they looked at was appetite regulation, and the impact was on the expression of appetite peptides. Now, in vivo studies demonstrated that the crude saponin fraction extracted from Korean red ginseng in rats led to a notable 20 to 30% reduction in body weight and decreased food intake in both normal and high-fat diet rats. So it means that it shows signs that not only is it going to lower the body weight, but it's also going to lower the desire for food, at least in the rat model. There's also mechanism as it relates to lipid metabolism, and we read a little bit about that earlier on. The anti-hyperlipidemic activity is what they looked at, and they found that ethanol extract of trilline or steroidal compound isolated from the machina rhizome demonstrated the ability to mitigate the high-fat diet-induced increased cholesterol levels in rats. Moreover, it restored the HDL and LDL levels to normal, indicating robust potential in anti-hyperlipidemic activity, which is a part of metabolic syndrome, because in metabolic syndrome, you not only have visceral fat deposition, but you also have high lipid in the blood, high cholesterol, and hyperglycemic instances, as well as high blood pressure. So this is going to really have a good potential in helping persons with metabolic syndrome. As we come to the end of our presentation, let us ponder a few things. Is it feasible to consider food as medicine? Can the inclusion of saponin-rich foods be integrated into treatment approaches for preventing, managing, or treating obesity alongside conventional therapeutic methods, including pharmaceuticals? Is there potential for using a saponin as an intervention in metabolic syndrome? Thank you so much for listening, and I'm going to ask that you continue watching as we have a summary video that ties everything together. Thank you.
Here are the references that I used to prepare this presentation.
Have you ever pondered the enormity of the obesity epidemic that our world is currently grappling with, or questioned if there might be novel, more effective treatments on the horizon? Well, you're not alone. Scientists around the globe are tirelessly exploring new avenues to combat this health crisis. And recently, they've unearthed some promising potential in an unexpected place, the plant kingdom. Delving into the world of botany, we find a group of plant compounds known as saponins. These naturally occurring substances have been a staple in traditional medicine for centuries, and modern research is now catching up to the potential that our ancestors recognized so long ago. Thanks for listening. Recent studies are focusing on the role these plant-derived saponins could play in managing obesity. But before we dive into the specifics, it's important to understand what saponins are. These are a class of chemical compounds found in particular abundance in various plant species. They're named for their soap-like qualities, having the ability to form a soapy lather when shaken in aqueous solutions. But what makes these saponins so interesting to scientists is their potential impact on our bodies. Preliminary research suggests that saponins could interfere with the normal process of fat absorption in our intestines. By inhibiting this process, saponins could potentially prevent the accumulation of excess body fat, thereby helping to control weight. Recent studies have shown promising results. For example, a study published in the Journal of Nutritional Biochemistry found that saponins extracted from the plant Camellia sinensis, more commonly known as the tea plant, effectively reduced body weight in obese mice. The saponins achieved this by inhibiting the absorption of dietary fats and cholesterol, suggesting a similar effect could occur in humans. In another study, saponins derived from the ginseng plant were found to stimulate the body's metabolism, increasing the rate at which the body burns calories and uses up stored fat. Weight control programs commonly incorporate elements such as dietary management, physical activity, and pharmaceutical interventions. The utilization of plant-derived saponins in obesity treatment holds significant promise, as emerging evidence indicates their potential benefits through diverse mechanisms of action. Both saponin-rich extracts and pure compounds have shown efficacy in inhibiting pancreatic lipase, modulating adipogenesis, and regulating appetite. Additionally, the inclusion of saponin-rich legumes in the diet presents a potential avenue for facilitating obesity control, points to another potential avenue through which saponins could help manage obesity. However, it's important to note that while these findings are encouraging, they are still in their early stages. More research is needed to fully understand the mechanisms through which saponins affect weight, and to determine the most effective dosages and forms of administration. To summarize, the key points about the potential of plant-derived saponins for obesity treatment are: saponins are naturally occurring compounds found in various plant species. Preliminary research suggests that saponins could interfere with the absorption of dietary fats and cholesterol, potentially preventing the accumulation of excess body fat. Some studies have shown that plant-derived saponins can stimulate metabolism, thereby increasing the body's calorie-burning capacity. Though research findings are promising, they are still in their early stages, and more studies are needed to fully understand the potential of saponins in obesity treatment. Saponins are susceptible to heat, and their quality diminishes, leading to a reduction in nutritive value during processing. The traditional processing method for beans involves thermal techniques like canning. In their 2004 review article titled "Saponins from Edible Legumes: Chemistry, Processing and Health Benefits," Shi et al. Investigated the impact of thermal processing on the characteristics and stability of saponins in canned bean products. The study revealed that during soaking and blanching, portions of saponins dissolve in water and are lost in the soaking, washing, and blanching liquids. Optimizing the thermal process has been proposed as a means to enhance stability and preserve saponins in canned bean products. This optimization holds value for advancing thermal processing technology in the food industry, improving the quality of bean products and consequently enhancing the associated health benefits. So the next time you're sipping on a cup of tea or enjoying a ginseng-infused beverage, remember you might just be partaking in a sip of science, a toast to the potential future of obesity treatment. And as we continue to explore the vast potential of our natural world, who knows what other health-promoting secrets we might uncover?